Hello and welcome to Mr. Queen Science as we start our first week of AP Biology Review. This week we will be looking at some of the core concepts from Unit 1, which is about the chemistry of life. So each week we will be looking at a different unit. The AP Biology curriculum is divided into 8 units of study according to College Board. Each unit has a different focus, but also a different exam weighting. So definitely pay attention to this when you're studying. Again, this doesn't mean that anything is less important or more important than anything else, especially in biology, which is a very integrative topic, even if it doesn't seem like that at first. By the time you get to the end of the course, hopefully you're starting to see how everything connects. This unit, although I don't see a lot of individual questions targeted at just these specific topics, they are absolutely foundational to the majority of the things you will learn about in biology and the majority of the things you will be tested on the AP Biology exam. So again, this week we are covering the chemistry of life, one of the 8 units recommended by the College Board for outlining the AP Biology course. And the major topics we're going to focus on within that unit are on the right-hand side. So tonight we're going to be looking at the structure of water and hydrogen bonding, the elements of life, and all 4 of those last topics can be summed up essentially as macromolecules. So we'll explore a little bit of the details about macromolecules. Again, where did this information come from? This comes from College Board. They actually have a gigantic 200 and something page document all about AP biology. I don't recommend that you learn that. It's more designed for your teacher or your instructor. However, there are some key pieces of information there. So I'll try to highlight those each week as we review each topic. You might also notice that there are these little color codes that kind of, again, these are aimed at your teacher. However, they do give a little bit of context of what's most likely to be tested, the style of question or the science practice that to see demonstrated. So for example, this one right here with the orange represents argumentation. So of unit 1, the one topic that's most likely to involve some level of argumentation, maybe you're going to need to use structure and function to defend an argument of why a certain molecule behaves a certain way or a certain property at maybe a higher level than the molecular level. It's most likely going to be from this unit at least based on structure and function. Now that doesn't mean that any topic isn't fair game for any style of question. It definitely is. But obviously, if College Board outlines this as part of their strategy of designing and teaching the course, there's a good reason to believe that this information can be considered valuable in some way. All right, so let's get started for today and jump right into our chemistry of life unit. Now, the first major topic in this unit is the structure of water and hydrogen bonding. Some of the key stuff that College Board wants you to know is that living systems depend on the properties of water. Specifically, they emphasize the importance of cohesion and adhesion and surface tension, which we'll get to in a little bit. But before we can dive into those properties of water, it's pretty important to analyze the structure. So water is a very important molecule for a few reasons. The first one is water is polar. And for those of you who haven't taken chemistry or you haven't taken it in a while, you might say, what the heck? They keep talking about this polar stuff. What does it mean? So here we're going to do a quick 1-minute breakdown of what it means to be a polar molecule. Now, in this molecule, oxygen and hydrogen are connected by these little lines. Those are called covalent bonds. They represent a sharing of electrons between oxygen and hydrogen. Now, if you remember your chemistry knowledge, covalent bonds are supposed to involve sharing electrons. However, if you've ever had that friend you go out and order some French fries with, and they might say like, "Hey, let's get 2 orders," or you say, "Let's get 2 order fries." Like, "No, no, I'm not that hungry. Let's just share." You're like, "All right, let's do it." And then you get the fries, and they eat like over half. You're like, "Oh my gosh, they did it again." That's kind of what's going on with the electrons in a water molecule. Oxygen is very hungry for electrons. We call that electronegativity. When an atom is hungry for electrons, it attracts more electrons. So oxygen sort of hogging the electrons in this apparently sharing relationship. What the result of that is at the overall molecular level is there tends to be more negative charges around the oxygen. So it tends to be a slightly more negative, and we represent that with this little delta sign and a negative. And the hydrogen side tends to be a little bit more positive. These are sometimes referred to as dipoles. They're not complete moments of polarity, they're not full positive and negative charges, but there is definitely some discrepancy in the charge in the molecule. There's definitely some amount of electric or magnetic polarity, which is why we call these molecules polar. Because of these negative and positive charges, they're going to interact with each other since we know negative and positive attract. So in this little diagram, it gives you a good picture of that, where we see the negative oxygen is having an interaction with the positive hydrogen of a neighboring water molecule. Now, these are interactions between different water molecules, whereas the covalent bonds are interactions within a single water molecule. So this process of being able to have interactions with other water molecules is referred to as hydrogen bonding. Hydrogen bonding is not a real form of chemical bonding like ionic or covalent bonding. It's more of just an interaction between molecules. But because the interaction is fairly strong, it's referred to as a hydrogen bond. And this gives water many of its unique properties. These hydrogen bonds give water a unique geometry in its solid form, and also are part of the reason why it has a lot of interesting properties such as cohesion, adhesion, which we'll get to in a minute. It's factors regarding its specific heat, and also its ability to act as a polar solvent in biological systems. That's probably one of the most crucial elements of water. So key misconception, don't mix this up. The covalent bond is between the oxygen and the hydrogen in the same molecule. They are sharing electrons. The hydrogen bond, on the other hand, is just an attraction between waters. I can't stress that enough. Okay, so a little bit more on polarity, especially how it applies outside of just one type of molecule, the water molecule. There's actually lots of molecules that can be considered polar. And what makes a molecule polar? Well, a polar molecule is going to have a discrepancy between an area in the molecule that's positive and an area in the molecule that is negative. Why does this exist? Because some atoms are more electronegative than others, which means that some atoms, if it's like a little tug of war, I'm going to try to draw on the fly, right? And this poor little guy over here is positive. He's not as buff. He can't hang on to those electrons quite as much. So because of that, it results in an unequal sharing. I got this greedy guy for electrons over here. So electronegativity is at the core of these bond polarities. So when we have a difference in electronegativity, we have a difference, we results in a polar covalent bond. Now you might ask, how am I supposed to know the electronegativities? I'm not even in chemistry. Well, even if you haven't taken chemistry, or even if you were in chemistry, electronegativities are essentially just values that you can use a table to look them up, but you can also just know some general trends about electronegativity. In general, elements in the top right corner of the periodic table tend to be more electronegative. Elements like fluorine, oxygen, nitrogen, and chlorine. For this course, we're really going to want to focus on when oxygen or nitrogen binds to something, that's going to tend to be an uneven sharing of electrons. One specific bond that involves two different elements that isn't really very polar is the CH bond between carbon and hydrogen. Although there is some difference in their electronegativity, it is not substantial enough to qualify as a polar bond. So typically, if a molecule has lots of carbon and hydrogen, we're going to consider it nonpolar. Another thing to consider is if we have two of the same atom binded to each other, then that's also going to be nonpolar. Think of it as a tug of war. You're right, oxygen is greedy for electrons, and so is the other oxygen. But if they're equally pulling on those electrons, neither one's going to kind of win and have the more negative side. Last thing, before we move on, bonds. You're like, oh gosh, how the heck am I supposed to do this? So what are the main types of chemical bonds you should know about for biology? Well, we talked about hydrogen bonds, but they're not really bonds. So we have covalent bonds, which involves the sharing of electrons. And then we also have something called ionic bonds, which is actually a transfer of electrons. Now that might seem similar to you on the surface, but what that really means is covalent bonds really do share the electrons. Neither one is electronegative enough to completely hoard it for itself. Now, an ionic bond, the discrepancy in electronegativity is so great that one of the atoms will literally rip the electron away. It's not sharing anymore, it completely stole it. And then, because it stole it, it became more negative. The other one became more positive. And now there's actually an attraction between the positive and negative charges. All right, well, let's keep moving forward as we talk about water. Now we've arrived at the properties of water, and all of these properties can be determined by the structure and particularly the hydrogen bonding and polarity of water. So the first one, and probably the most important one on this list, is cohesion and adhesion. So cohesion is the attraction of water molecules to other water molecules. So I would say water to water, but in fact, it could be any kind of molecule. So let's just go ahead and say attraction to self. That's not some narcissistic thing. Attraction to self just means that cohesion is describes when water molecules are hydrogen bonding to other water molecules. And this is going to generate things such as surface tension. What else does it cause? Yeah, that's like the main thing that you should know that it generates is surface tension. Essentially, water sticks to itself. So when you see a little bead of water form like on a tabletop, if you spill water, it doesn't spread perfectly flat out everywhere. It creates a little bead. You see this on your car windows in the winter when it's raining as well. On the flip side, you have adhesion. So that's attraction to another type of molecule. Okay, so this is when water has interactions with other molecules. So you can jump in a swimming pool and you get out, you're covered in drops of water. It's not like the water just slides right off your body. So that's an example of adhesion. And adhesion is one of the main reasons that things like water can move up through the roots of a plant. Water gets attracted to the insides of these different cellular passageways that essentially allow it to move up through the plant. It's the kind of sticking and crawling its way up the walls. There is more to it as well. There's transpiration and things, but that's not the topic we're talking about right now. So I'll get back to what we're discussing. Another important property of water is its unique density. Water does not, I mean, when water freezes, right? So solid water, ice. Okay, so ice, most solids, when they become a liquid becomes a solid, it becomes more dense than the liquid, so it will sink to the bottom. Ice is unique because it floats on the top of the water, which if that didn't happen, our oceans would freeze from the bottom up and life would have never evolved in them, which would eventually never have led to life on land. All right, so the next thing is it's a biological solvent. Because it is a polar molecule, other polar molecules are going to dissolve in it. So you might hear this phrase from chemistry of like dissolves like. Okay, so water dissolves polar compounds. So all these different ions and a lot of polar molecules dissolve in water, and all of biology essentially happens in water. Biochemistry exists within a solvent, which is water. You've probably heard your body's 75% water, all these different things. Your cytoplasm is water, your blood is mostly water. So it's very important as its ability to act as a solvent. And finally, there is a high specific heat when it comes to water. What does that mean? So that's specific heat is basically a measure of energy or heat. Whoops, energy or heat to raise the temperature of 1 gram of a substance by 1 degree. So water, it takes a lot of energy to increase the temperature, increase the average kinetic energy of that substance. That's actually really good for biology and ecology because it holds heat. So at night, water doesn't become freezing cold. Water will actually hold heat in. If you've ever been somewhere near the water at night, it's actually a little bit warmer than if you were out by the mountains. The soil is going to get colder more so than the water. On the flip side as well, also water doesn't just evaporate away when it gets too hot. It takes a while to raise the temperature of water. Again, another important thing. Now, if any of these concepts are confusing to you, I'm not usually trying to refer you out to other videos, but check out the TED Ed video on H2O. It's a really good overview of water. It hits all of these properties and the polarity aspect in a short 3 to 4 minute video with some good visuals as well. So check that out if you're still having trouble understanding this concept. Okay, so let's go to our next topic. Oh, as a little segue before we jump into looking at the next thing, which are the chemicals of life, I wanted to briefly touch on functional groups because it does have a relationship to polarity. So a lot of times on the AP exam or in your AP bio class, you're going to see these large organic molecules. Organic means based out of carbon. And this is an example of just a small fragment of a protein. This is only 6 amino acids of a protein. Some proteins can be thousands of amino acids long. So you might say, how the heck am I supposed to know anything about the polarity of this molecule or how it's going to interact? How can you expect me to learn this kind of stuff? I'm not in college yet. Even I'm not in organic chemistry. Well, all we really need to focus on here are the functional groups. These are small groups of atoms that have unique properties that can actually translate into properties for an entire larger molecule or a portion of a larger molecule. So most of the functional groups that you should know about are polar. There's a very nice diagram in the Campbell textbook and other places online that outline all of these molecules and give you more information, such as examples and structures and things like that. So I'm not going to go through every one besides reading them off, but hydroxyl, carbonyl, carboxyl, amino, sulfohydryl, phosphate, they're all very polar because they do have that discrepancy in electronegativity. So OH, as we've seen before, it can create polar bond, hydrogen bond. It's a polar molecule, has a polar covalent bond. The only one on this chart that's not polar is the methyl group. This is simply a cluster of carbon with 3 hydrogens bound to it. So in general, if you see lots of carbons and hydrogens in a molecule, it equals that it's non-polar. If you see nitrogens and oxygens, that tends to going to be, or phosphates, that tends to be a more polar functional group. So it's going to give that part of the molecule polar properties. This is absolutely crucial to understanding. They're not going to ask you a question point blank, like, what is this functional group and is it polar or not? But when you get to these bigger concepts relating proteins and nucleic acids and going from the molecular level up through the cellular and organismal level, of biology, you're going to see that this polarity is essentially at the core of all of the different properties that can be derived from the structure and function of these molecules. So again, you don't need to memorize how to read every single thing, but do be familiar with these functional groups. It will make, even if there isn't a question exactly about them, it will make understanding things in the future units much easier. All right, so our next major topic, as outlined by College Board in this unit, are the elements of life. And let me double check what is the exact thing that they're looking for here. Let me read it off. So these elements of life, organisms must exchange matter with the environment to grow, reproduce, and maintain organization. So we're going to dive into that topic a lot more in week 8 when we look at ecology. However, there is an application of that to this concept of elements of life. And what is meant by elements of life is that there are some elements on the periodic table that are much more important to biology than any of the others. So you might understand now why some branches of science kind of look down on biology, because this is like the kind of stuff we do to the periodic table. That's all we want. This is good enough for us. This is all we really need. And to be honest, all those white elements too, we could probably just like forget about them. So according to this table, we have these elements highlighted in the deep red color. These are going to be our essential atoms or essential elements in the in biology. So we've got sulfur, phosphorus, oxygen, nitrogen, carbon, and hydrogen. These are going to be found in all living things that we know of on Earth. The ones in pink are needed in trace amounts. There's not that, like, they don't need to be a lot of it, but there does need to be a little bit of it. Most of these serve a role in facilitating some kind of biochemical process, acting as a catalyst or acting as a cofactor to a protein enzyme. The ones colored in gray, chlorine, potassium, calcium, magnesium, and sodium, they are essential as well. However, they're usually more involved in their ionic form as opposed to a molecular form, and they're not as essential as the sponge or CHOMPS elements. So just make sure you know that. A good way to remember it, like I've said before, is sponge or CHOMPS. They're probably not going to ask you point blank, like, is phosphorus an essential element? That's not, if you've been studying at all, you know that that's not what the AP test is like. But they might bring up something about, how would we even say this? There is applications of it in later units when we see some of the experimental design of different molecular biologists. They oftentimes take advantage of this fact that there are these essential elements, and some molecules in biology contain certain ones, and some molecules contain others. All right, so moving on, let's move ahead. We will touch back on this whole chomps thing and sponge, like where do these elements of life appear when we touch on macromolecules towards the end of the video, which we're already at. So this says structure and function of biological macromolecules. Now, according to the AP guidelines, they do break this into three separate subtopics. I think it's like an introduction to macromolecules, the properties of macromolecules, and the function of structure and function of macromolecules. We're going to kind of just talk about all of them at once. Something else that I would like to highlight here are these exclusion statements that you can see in the top right corner. The AP College Board materials have specific exclusion statements on some of these topics so that you basically know that what is and what isn't on the test. Now, they aren't that clear always about all of that stuff, but these exclusion statements are the gold standard of knowing what's not on the test, at least. Now, if you read through all three of those, they essentially say something very similar, that the molecular structure of specific nucleotides, amino acids, carbohydrate polymers, and specific lipids are not going to be on the exam. So you don't have to know the exact type of linkage in cellulose versus starch. You don't need to know the structure of the amino acids, like by atom, but you should know some of the general information about. Okay, so our first thing that I say that's worth mentioning, so we can kind of use it as background information for the rest of our discussion of macromolecules, is the difference between polymers and monomers. So polymers in general, it's not just in biology, it's in chemistry as well, is essentially a molecule that's made out of many small subunits, kind of like a chain. It has repeating subunits over and over again. And if you look at this word, poly means many. So this sort of tells you that, yeah, this has many links in the chain. A monomer is one of the single links in the chain of a polymer. So 3 out of 4 of the macromolecules we look at today are going to be polymers built of monomers. The other important thing we want to know going into our discussion here is two types of important reactions: dehydration synthesis reactions and hydrolysis reactions. Dehydration synthesis reactions involve, well, if you break down the word, dehydrate, so somehow removal of water, and synthesis, which means to build something. So this is the process used to build polymers from monomers, and it involves the removed molecule in the process. So you're like, wait, where is the H2 molecule coming from? Well, it will usually take some hydrogens, oxygens, and hydrogens from two different monomers while they're joining them, and water will exit, and the byproduct will be that they're linked. You don't need to know the exact mechanism of that or the exact organic chemistry of it. Just know that dehydration synthesis builds polymers through a reaction that removes a water molecule. And actually, dehydration synthesis will be used in many places in biology if you look at the biochemistry of it, including like ATP, breakdown, and stuff like that, and production, and breakdown. On the other hand, you have hydrolysis. So these are kind of like opposites. They're like partners. If dehydration synthesis builds polymers, hydrolysis is going to break it down. Again, hopefully you're starting to see some patterns in the words you study. Hydro means water. Lysis means to cut or break. So hydrolysis means using water to break the molecule. So this is going to be to break polymers back down into monomers, and it uses H2O to do that. It's going to add a hydrogen to one monomer and an OH group to the other. All right, so now let's dig in a little bit deeper into the different classes of macromolecules, and I'm excited to show you what I think is a very important study strategy and skill for AP Biology. Okay, so especially if you're not cramming, if you're actually trying to follow through with this week by week, set up a little bit of a study plan. This would be one piece of advice I would give you, is to essentially, I know no one likes rewriting things, but it's not really about the rewriting part as much as the reorganizing part. So I find graphic organizers of all kinds, whether those are tables, charts, word webs, Venn diagrams, labeling exercises, any way that you can organize the information requires you to manipulate it in your mind and to adjust the way that it fits into the network of associations, which we call like knowledge or whatever. So again, taking information and reorganizing it in a new format really helps your brain understand it. The average person, I think the study from a while ago says something like about the average person requires about 8 exposures to information in order to really start integrating it into their long-term and or at least midterm. So what that means is you hear it in class, you watch a video, you do a lab, you take notes, you reorganize your notes into a table, maybe you make some flashcards. Every time you work with the material in a different way, it's getting more and more connections in your brain, so it's sticking with you. Okay, so if you do have the time, if you are a little bit ahead and this isn't, you're not watching this minutes before the AP test, I hope. This is one way that I like to organize information, especially if there's an element of compare and contrast here, where I want to see what are the differences and similarities between different concepts in biology. So I'll break this down a little bit. We're not going to go through all the details of this, but just hopefully this is something you can do on your own as well. It's going to be more powerful than just watching me do it. So what elements are in carbohydrates? Well, it's usually CH and O. Same thing as our lipids, CH and O. There's some special lipids that also have phosphorus in them. We'll get to that in a minute. Proteins usually have CHO and nitrogen. Very important part of proteins is nitrogen. And nucleic acids have your CHO and nitrogen, but they also have phosphorus. Sometimes proteins can have sulfur as well, but again, it's not always. Okay, so another name, as we start to get into the monomer portion of this, another name for carbohydrates, especially our polymeric carbohydrates, is going to be a polysaccharide, which means many sugars. The monomer then of a carbohydrate is called a monosaccharide. Okay, so that means single sugars. So you connect many single sugars together and you get a polysaccharide. Now, usually when we mention carbohydrates, we're thinking about polysaccharides, although technically monosaccharides are carbohydrates too. I know it's like a weird nomenclature thing. Just understand most carbohydrates are polymers built of these monomers, monosaccharides. Lipids do not have a monomer. We'll get into lipid structure in a little bit. Proteins, they do have a monomer. Those are called amino acids. Hey, and flashback to our functional groups from a few slides ago. Okay, amino acids have two really important functional groups. They have a carboxyl group, also known as a carboxylic acid, and they have an amino group, also known as an amino group. So when you combine those, amino acid, you get, the building blocks of proteins. Whoops. Okay, so building blocks of proteins are called amino acids. They involve nitrogen, of course, for the amino group, and they also have a carboxylic group. And finally, nucleic acids. They have a monomer called nucleotides, probably the most complex of all the different monomers of these molecules. Okay, I was going to go over structure, but I think we're going to go over each of those one by one. So instead of structure, let's talk a little bit about polarity. Again, when you make a chart like this, you could make it way more rows. The more things you put in this grid format, you have all the data right there. I mean, you might find a table like this already made online, and that's really helpful too, but there is some value in it of actually taking your notes, making sense of them, and sorting them into a new organization. It forces you to work with the information. All right, so polarity of these guys. Carbohydrates are generally polar. They've got a lot of oxygen, a lot of functional groups. Lipids, they're the odd man out, as you can tell. They don't have a monomer. They're generally non-polar. That doesn't mean that there's not any polar groups in lipids, but we generally think of them as non-polar since they are majority C's and H's. There is some O's, but it's majority C's and H's. Proteins are definitely polar, and nucleic acids are definitely polar. Maybe the most polar. They have a very strong negative charge due to the amount of phosphate. And again, if you look back at those functional groups, phosphates, not just phosphorus, but phosphates, have a very strong negative charge, which is going to make it very polar. Okay, so what is the function of these guys? Oh gosh, so there's a lot more than I'm going to write right here, but of course, hopefully you know the main reason that's like you might carbo load or eat pasta or sugar before a sporting event is going to be energy. It's an easy, accessible source of energy for most organisms, whether that's going to be using fermentation, if there's no oxygen present, or if you're a prokaryote, or using cellular respiration to break down glucose into ATP. So energy is probably, I'd say, it's like the main energy molecule. The other thing is structural. In certain organisms, carbohydrates can provide a structural element, such as building up the cell walls in plants. Cellulose is a carbohydrate, and it builds the tough, rigid structures of cells and cell walls. I mean, there are also other little things for carbohydrates. They can act as essentially sort of like signaling molecules as well. We'll get into that in Unit 4 about cell communication. Now, lipids, what's their main function? Okay, so there's a lot here. They can be used for energy. It's usually more of a storage or like long-term function. I think A lipid probably contains more energy within its bonds, but it takes more work and a process to essentially extract that energy. So it's more of a long-term storage molecule. What else do we know about lipids? So they're also in things like hormones, so they can be involved in signaling or communication between cells. And then finally, they can be used in the phospholipid membrane. So I'm going to say structural again, and we'll explore what the phospholipid membrane is a little bit today and in the future as well. Now, proteins. Proteins do it all. I don't even know what else to say. I could list like a million things, but they can be enzymes, which means they're speeding up chemical reactions and helping them out. They can be involved in transport at the cellular level, moving things in and out of the cell. They can also be involved in structural elements of the cell, which could be at the cellular level, providing that kind of structure, or they could also be providing the structure of that leads to different kinds of traits. So all of those things are going to be done by proteins. I mean, they can also be regulatory, which we'll see. All in all, proteins do so much. If you ever run into something in biology and it's like asks you what it is and you're not sure, I'm not telling you it's always going to be right, but protein's a pretty good guess. Most stuff's made out of proteins. And then finally, our nucleic acids. These guys are DNA and RNA, which have unique functions, to be honest with you. DNA is more of like it's the genetic info, but it kind of just stores it. It's not actually doing much. It's just hanging on to it, making sure it's being copied accurately, and essentially containing the info that will lead to the production of proteins, which defines a lot of aspects of an organism. Now, RNA is more involved as how I don't know how to say it is. I'm going to call it a tool of protein synthesis. I think that's the best way to put it. So whereas DNA is holding the information, RNA is actually a molecule that has the ability to perform functions such as carrying some of the messages that's in DNA, helping build proteins, all kinds of stuff. There's tons of things we can talk about in RNA when we get to that unit. So we're going to move forward and talk about each of these macromolecules more specifically. So we're going to start with carbohydrates. This diagram shows you a little bit about the structure of these different carbohydrates. Again, it said in the exclusion statement, you don't have to memorize like what's the structure of fructose or what's the structure exactly of glucose. Now you should know it's made out of carbons, hydrogens, and oxygens. You should understand some of the bonds that are involved in that molecule, but you don't have to draw any of these out. Same thing with you don't need to know the difference in the linkages between cellulose, starch, and glycogen you can see in the bottom corner, but do know there are differences. So all three of those molecules are made entirely of cellulose, but by the way that they're bonded and the types of branching in their bonds as well, they perform very different functions for different organisms. Cellulose is more used as a structural thing, whereas starch is more of a storage molecule for these sugars. All right, so that's like I'd say that's most of the kind of core stuff you got to know about carbohydrates. Again, the actual structure is going to dictate so much about its function. The way that these things are bonded together is going to be the difference between what's a structural carbohydrate and what's a good energy storage carbohydrate and what's a carbohydrate that's going to be very accessible and easy to use for energy, not to store, but to access energy. So a big concept throughout all these molecules is that their structure is going to play directly to their function. Okay, our next major class of macromolecules are lipids. Look at all these crazy, beautiful structures. So at the core of most lipids are fatty acids. Now, it's not always the case, but most of them have some element of a fatty acid. And a fatty acid is a long chain of mostly carbons and hydrogens that is going to be part of other lipids. The most common type of lipid is called a triglyceride, and a triglyceride has this consistent structure of a glycerol molecule that has the ability to make three bonds with other fatty acid chains. So different fatty acids can bind with this glycerol. Based on the type of fatty acid, it's going to generate different properties in that triglyceride. Now, saturated fatty acids and unsaturated fatty acids, they are slightly different. The difference here is that unsaturated fatty acids have double bonds present, which result in a sort of kink shape, a bent shape. And this again will have effects on the behavior of that molecule and what it's used for. So they don't compact as tightly, therefore they don't have as strong of intermolecular interactions, so they tend not to be solids at room temperature. They tend to be liquids at room temperature. Now, a special case lipid is the phospholipid. So it has the glycerol molecule, it has the fatty acid chains, but it also has on one of the glycerol binding points, it has a phosphorus group or a phosphate group, which we know is highly polar. So phospholipids are unique because they're not hydrophilic, meaning they're polar and love water. They're not hydrophobic, which means they're nonpolar and they hate water. Essentially, they are, I guess you might say, ambiphilic. I think that's the word, which is that they have properties of both. So the same idea behind a phospholipid is the same idea behind soap as well, where the non-polar region of the soap molecule can bind to the dirt, and the polar region will get bind to water that sweeps it away. Phospholipids are important at constructing the phospholipid bilayer of the cell membrane, which we'll talk about in much more detail in the next video. One final class of lipids is the steroid. Steroids are unique in that their structure is very different. It's a 4-ring structure, mostly carbon and hydrogen, and it is based off of the general structure of cholesterol. It's generally used as some kind of signaling molecule in the body, and because it is nonpolar, has some unique properties, such as being able to cross the cell membrane. All right, moving on to proteins. Proteins, like I said, is probably the most critical of these to understanding the process of life, not because it holds genetic information, but because of its versatility in doing so many things in the cell. So there's 4 levels of structure in proteins, which are worth knowing. The first one is the primary structure, which is literally the sequence of amino acids that make up that protein. The secondary structure is when the how would you say, the backbone of the molecule. So that's this, not the, if you remember the image we saw before, we saw that there was kind of like things branching off these amino acids, different kinds of side chains. Now, that's not what secondary structure is about. Secondary structure is about the actual atoms and molecule that's holding and the bonds holding things together, how they interact. And they usually interact in one of 2 ways. They form something called an alpha helix, which is like a spiral type shape, or they form something called a beta pleated sheet. These interactions are not due to the side chains, they are due to hydrogen bonding between the backbone elements of the molecule. So tertiary structure, now we can talk about how our side chains interact with one another. And since we know things that are non-polar like this stick together and things that are polar like this stick together, that's really what's going to dictate how these interactions occur. But what this manifests into at the 3D level is a unique 3D shape, which provides the protein its structure, which of course is connected to its function, whether that's the active site in an enzyme or a specific shape that allows it to be very strong or provide some kind of support or structure to other elements of the surrounding tissue. And finally, the quaternary structure is when every once in a while, not every once in a while, but not all proteins have this, is what I wanted to say, is that multiple tertiary units can interact together to create a larger protein complex, so to speak, that contains these separate polypeptides. Polypeptide is just another word essentially for a protein or a series of amino acids that have been linked together. So that's the structure and function of proteins. Again, all of its function at the deepest level is embedded in the unique sequence of its monomers, which result in specific type of secondary structure, which then results in tertiary structure, which may result in quaternary structure. But eventually, as we go up, it's going to affect the way the cell manifests itself and the way the organism manifests itself as well. All right, we're almost done. We're at our last topic for today, which is nucleic acids. So this is our 4th and final macromolecule to discuss as we talk about biochemistry. Now, nucleic acids, I usually like to teach them in Unit 6, which is more about gene transcription, translation, expression, all that kind of stuff, since DNA is so central there. However, AP does point out to teach it in this unit. Your teacher may or may not have done that, that's fine, but just for the sake of being consistent with their materials, I'm going to discuss it briefly tonight. So there's 2 main types of nucleic acids, DNA and RNA, and they're built, if you remember what we mentioned earlier, they're built from monomers. And the general structure of a nucleotide monomer is going to be like this. This is a sugar, this is a very polar phosphate group, and this is what we call a base. Specifically, you might call it a nitrogenous base. It means it has molecules that involve nitrogen inside of it. So that general shape is going to be the same or similar in both RNA and DNA. The big difference is a couple things. First of all, the sugar in DNA is called deoxyribose. So that sugar is called deoxyribose. And second of all, in RNA, it's called just ribose. So the slight difference in the sugar is part of what makes these nucleotides different. Another difference that they have is that the types of bases that can be present. In DNA, we see these four: thymine, cytosine, adenine, and guanine. And however, on RNA, we see some of them are similar, but instead of thymine, we see uracil instead. Again, in a future video on Unit 6, we'll get into some of these details and why they're important a little bit more. So that's what's going on at the nucleotide level, but let's look at the rest of the structure. So the DNA molecule, as you can hopefully see, is a double-stranded molecule. It has two kind of chains, so to speak, or of these. However, RNA is a single-stranded molecule. So that's one important thing about it. We already talked about the sugars that are present. And I guess the last one is that DNA tends to have this main form is the main form of it. It looks like this spiral staircase. R can look like that picture, but it can also do some pretty unique stuff. It can kind of bind and interact with itself, and it can form a lot of different unique shapes. As you get later into the course, you'll understand that there's different types of RNA: mRNA, tRNA, rRNA, small nuclear RNA, all kinds of RNAs, and they all serve different functions. Okay, so which leads us to the next point, which is the function of these molecules. DNA, as we just said earlier, is basically the storehouse of genetic information in the cell, whereas RNA plays more of an active role of actually seeming more to facilitate the process of getting the information from the DNA and transcribing it and translating it into the proteins, which will essentially provide the organism with the structure and function that will make it unique. The last thing to mention is the location. In DNA, the location is usually found in the nucleus, and in RNA, it can be found in the nucleus, but it can also be found out in the cytoplasm. It can also be found at a ribosome. You're like, wait, why is it at a ribosome? Well, heck, a ribosome is actually made out of RNA. So that's definitely one place you find RNA. Last thing I want to mention that hopefully you can see in the DNA molecule, it's not as evident in the RNA molecule, is the phenomenon of base pairing. So in nucleic acids, what scientists have discovered is that certain molecules tend to pair up with one another. And in this case, it's going to be thymine and adenine tend to pair up, and cytosine and guanine. Okay, so we call this base pairing. And within the DNA molecule, unless there's some kind of error, we should always see this kind of base pairing. Now, RNA, you're like, well, it's not double-stranded. Doesn't really matter. It can still interact with other nucleic acids, such as DNA, other strands of RNA. It can even interact with itself. Its version of base pairing is a little bit different. In its case, uracil and adenine bind, and cytosine and guanine bind. All right, so there you have it. That's like the main things about nucleic acid. Again, like proteins, like lipids, like carbohydrates, the structure, the specific sequence of different components, whether those are monomers, whether those are fatty acids and glycerols, whether those are amino acids, or whether those are nucleotides, the specific pattern or sequence will determine some of the larger properties of it, which can be in turn translated into describing the larger properties of a cell or organism. So that's unit one. Again, it's probably one of the easier, I feel like the easier units. A lot of students are like, oh my gosh, I did so bad at the beginning of the year. Well, heck, it was a brand new class. This might be your first AP. Hopefully, when you go back to study this material, you realize that it's not as hard as you remember at the beginning of the year. You've learned so much more biology that it sort of makes this stuff more understandable and easier to grapple with. In terms of questions, there aren't too many questions that directly address Unit 1. There's like a handful, I'd say like a couple per year's multiple choice section, and maybe one element directly found in the FRQs. However, these concepts are absolutely foundational. You are going to be coming back throughout the rest of the course. So don't just ignore biochemistry. It's not that hard. There's only a few major concepts, and it will apply to your study throughout the rest of the course. So I hope you found this video helpful. If you did find it helpful, please like and subscribe. If you didn't, I would love to hear your feedback of how it could be better and what you might need. Again, if there's ever, for this channel in general, especially while we're small, if there's ever a topic that you'd like to know in greater detail or something that just doesn't make sense, whether from my video or from your teacher's explanation, let us know and we would love to make a video about it and kind of help you out at that individual level as you try to make your way through these AP courses. So again, thank you for tuning in, and until next week, we'll be working about cell structure and function. I'll see you then.